Right, this video is a review for the EOC, so it's EOC review Unit 1. Now I know I said I wouldn't do a video on Unit 1. However, after thinking a little bit more about it, I thought there are a few things that I would hate for you to miss just because you simply just forgot them. So let's talk quickly about a few of these things. One of this, what is the scientific method? And the scientific method is a process by which scientists do things, and it begins with observations and questions. I see a bird in a tree, and that bird every 5 minutes will fly down and then fly right back up. Well, what's the natural question? Well, I observed it first, and what is the natural question? Why does it do that? And so at that point, we make what's called a hypothesis, which is a guess as to why that happens. This is your projected or your predicted answer for the question that you asked. Well, I think the bird swoops down every 5 minutes because it sees food every 5 minutes or whatever. I don't know. Just make something up. Well, this is your hypothesis. So what do you have to do with that? You have to test it, and that is where you have the experiment. And in an experiment, you have certain variables that have to be tested. You have the dependent variable and the independent variable. All right, and the dependent variable, this is also called the responding variable. This is the variable that you are going to measure. This is the variable that you are going to be looking at in this particular experiment. It is always tracked on the y-axis. Like the amount of time between the birds going up and going down, or the amount, just do something that we've done in class this year, the number of bubbles that I see come up when I'm shining the light on the leaf disc, or the amount of time that it takes for the leaf disc to float to the top, or whatever that is. This is what you're actually measuring in the experiment, whereas the independent variable, this is what you're basing your measurement off of. This is the variable that you are personally manipulating. All right, the amount of bubbles that go up, which would be the dependent variable, over the course of 5 minutes. Well, this would be on your x-axis. You know, you would have a graph, and on the bottom you'd have time, and on the y-axis you'd have bubbles. And then at 1 minute you'd have it here, at 2 minutes here, 3 minutes here, 4, 5, 6, so forth, and that's where you draw your line. All right. Now there are certain other variables that have to be figured in here, and this is called your control. Now there are control variables, things that you're keeping constant in an experiment. Consider the leaf disc experiment that we've done. The things that we've kept constant is all of the water was the exact same. You use the same kind of cup in every different experiment. You do use the same amount of water in every different experiment. You use the same kind of plant. These were things that you controlled that kept the experiment nice and even. But you also had a control group. In the control group, let's say you were doing an experiment. Well, how many bubbles come up if I shine the light on it? And maybe the red line represents the line, the light. Well, we would also need to have a control group that said, well, it's the light that's causing the change. And so we would have another group that didn't have any light shining on it, and maybe we would represent that line here. And we'd be able to see then the difference between the control group and the experimental group. All right, so those are a couple terms that are easily missed and easily forgotten. A couple kinds of data that we talked about in class, real quick. We talked about qualitative data and quantitative data. And this is easy. Quantity, if I ask you what is the quantity, you're always going to give me a number. So quantitative data is always a number. It is how many feet is it in a mile, or how tall is that building, or how many times did something happen, or whatever. Qualitative data is the like quality of something, or the an immeasurable quality. Well, how bright is the sun? Well, we could do a actual measurement of that in numbers, but we could also just say it's really bright. Or how blue is it? It's really blue. Or that guy is tall, which is a completely different thing than saying that guy is 6 foot 3.5 inches. All right, one is a quantitative judgment and the other is a qualitative judgment. And another thing that science uses is something called inference. Now, inference is a logical conclusion based on prior knowledge or experience. I walk into a lake and I see that near the shore, there are bubbles, like little swirly bubbles coming up in several parts near the shore. Well, I know from prior experience that those represent some sort of living thing underneath the surface, like a turtle or a fish or something like that. Well, I can logically infer that there is something alive causing those bubbles to be made. Now, that isn't necessarily the case, but I can logically infer that, and then through further experimentation and gathering of data, I can then prove that inference correct. But inference is something that science does in order to make logical conclusions and in order to continue the work. So, another thing we talked about is measurement systems. We talked about the English system. This is our system of measurement, also shared by the countries of Liberia and Burma. We're in an elite group there. And these are things like feet, yards, pints, gallons, acres, etc. And we do not share that with most of the world because most of the world uses the metric system. Why do they use the metric system? Because it is in multiples of 10. And it could be that you're asked to do some sort of metric conversion. Now, we did these really early in the year, and some of you guys really did well, and some of you guys did not do well. And this is where we had these sorts of words, centa and desa, and I need to get smaller here as to not run out of room. And then we have the standard unit here in the middle, and then you have deca, hecto, and kilo. Now, this goes from, this is a 1/1000th, 100th, 10th. This is a 1. This is times 10, times 100, and times 1000. Now, the way that I do this is I'll actually turn it around backwards. Why do I do that? Because it makes it nice and simple when we start moving decimal points. And so I'll actually flip this thing around where I'll put the kilometers here, H here, DA for deca. And I'm using meters here, but I could just as easily be using liters or grams. And I'll just demonstrate that real quick. This is liters, obviously, capital L, and grams. All right, and I use this scale in order to convert things. So how do I do that? Well, let me take one here and let's work with it. So let's say I have 1 gram, and I want to see how many kilograms that is. So obviously I'm just going to change out these M's for G's. And how do I do that? Well, I'm going to start here in the middle at the G. And I'm going to go to the K. Here's the G, here's the K. And how many decimal points am I going to move? I'm going to move 3, 1, 2, 3, which way did I just go? To the left. And so for that, when my 1, where's the decimal point on a 1? It's right here, like as in 1.0. All right. I'm going to move that 3 spaces to the left. 1, 2, 3. Now what goes in these blank spaces here? Zeros. So 1 gram is equal to 0.001 or 1/1000th of a kilogram. Let's do another one. All right. One maybe a little more complicated here. Let's say 3.2 centiliters. That's not more complicated. Let's do hectoliters. What is a hectoliter? 2 centiliters. Okay. So how do we do this? Well, we're going to start at the H. This is L. We're just switching over letters here. We're going to start here down here at the H. And which way do we have to go? We have to go to the right. Well, how many spaces do we have to go? We have to go 1, 2, 3, 4 spaces to the right. So I'm going to do that. 1, 2, 3, 4. What do I put in these blanks? Zeros. What is that? 32,000 centiliters. Now, basically what's happening here is when I go to the right, I'm multiplying, so my number should always get bigger. When I'm going from a larger unit to a smaller unit, my number should always get bigger. If you tell me that 0.00032 centiliters, which is about the amount that you could fit in the end of your fingernail, probably even less than that, much less than that actually, is the same as 3.2 hectoliters, which I don't know, is like several hundred 2 liters of Coke, then you're crazy. All right, so make sure if you're going from larger to smaller that you're multiplying. Well, then what would we think we're doing if we're going from smaller to larger? Obviously, we're going to be dividing. So when you go smaller to larger, you are dividing. So the number should get smaller. This is a way to check yourself. So metric conversions, it is not outside the realm of possibility that you would see one of those. So, theory. This is another thing that we need to talk about here, this word theory. This is one of those words that is the most confused words in science because we often use the word theory in our own common vernacular as in, well, I have a theory about this. What is my theory? Well, I'm using that word as a guess. Well, really, what that word means is I have an explanation for something. The word theory almost invariably the definition for it will include the word explanation. What is a theory? It is an explanation for something that we see in science. And how do we come about that explanation? Through repeated experimentation. Let's use an example. Well, the theory of evolution is often downplayed as it's only a theory. But it's been shown over and over again through experimentation and through evidence. The theory, another theory that no one ever says it's just a theory, is the germ theory of disease. It's not just a theory. We believe that germs cause disease. We believe that bacteria and viruses cause disease, and this has been proven through experimentation, and this is how we explain sickness, through the germ theory of disease. So do not get theory and hypothesis mixed up. Very simple mistake. One last thing is the characteristics of all living things. This is one of those questions that sneaks up on you and you're like, oh no, I thought I knew that. Now, what are the characteristics of all living things? All living things are composed of cells. All living things reproduce. All living things have a genetic code, or DNA. All living things grow and develop or change over time in a this lifetime kind of sense. All organisms obtain and use energy. What do we call that? That's called metabolism. All organisms respond to stimuli. All organisms maintain stable living conditions. What's our catchword for that? Homeostasis. And lastly, all living things as a whole, as populations, change over time or evolve. So that's it for Unit 1. I don't suspect there'll be anything else covered. Maybe a few other uh, things that are common sense, but for the most part, I believe these are all the things that might have tripped you up on Unit 1.